All right, well, let's get started. Welcome to our first in a series of 10 webinars that we've titled, as you can see, Amplifying the NM Community Voice. As you will hear today, these sessions are meant to forge deeper connections to our community. Historically, there are many cases where patient advocacy groups have positively influenced the direction of research to apply their efforts directly to the needs of the population being served, and not necessarily towards the needs of science at the moment. We are excited to go on this journey with all of you, and we hope that we'll attract even more community members to get involved with our team that you're about to meet. Next slide, Lynn. So first off, I want to start off with some, we'll go through some introductions of the team members. We'll talk through the foundation building strength initiatives and our vision. We're going to talk about the tools to be able to apply and amplify the NM voice. We'll review our recent survey results. Thank you for those who have participated. We're going to let you know, the community know, more about how they can get involved. And then Dr. Alan Becks and Casey Giannetti are here as sort of principal investigators of NM in the world to be able to give you an overview of what the current state of NM is today and where we hope to go in the future. All right, we can see a picture of me there. I've definitely added some gray hair since then, so I may have to update my picture. Patty Mitchell has been a board member and has a daughter with NM. Has been our treasurer for a number of years as well. Lynn Byer, another board member, again with Greta, who you'll meet down in the bottom right corner. Both Patty and Lynn are important community members, community voices for us at the foundation. Dr. Gus Japolsky is our scientific director and project lead. Dr. Alan Becks, who I mentioned a moment ago, and his teammate, Casey Giannetti, will be here as well. Sarah Foy plays an important role today as our outreach coordinator for the PCori Project. And Stacy Cossette, not only as our program coordinator, but as our data control officer. Jen Tozer will be behind the scenes mostly, and she serves as our development director. Terry Selicki, our awesome grant writer who was able to get us this award with the help of many others. And then Greta Byer, our newest addition to the team, is our current youth ambassador. So I'd like to hand this off to Sarah Foy, our outreach coordinator for the PCori Project, to explain a little bit more.
Hi, thank you for the introduction. It's really nice to be part of this team. You may have seen me at past family conferences or online because my family is also affected by a rare muscle condition that impacts one of the large proteins that controls muscle function, and it's very similar to Nemelin myopathy. So it's natural that we have this connection or maybe met each other in the past. So I also have that understanding of what it's like to live on the roller coaster ride of life with congenital myopathy. So I'll be serving as the outreach coordinator for the PCori Award, which stands for, it's a little bit of alphabet soup, but the Patient-Centered Outcomes Research Institute, or we call it for short PCori. And I just want to congratulate the foundation on the PCori Award, and also, yes, very exciting, and also the grant from the Chan Zuckerberg Initiative. Just a brief summary of what is PCori and what is this project is that the aim of the PCori Project is to build an effective and lasting partnership among stakeholders, which involves you, of course, to drive forward patient-centered research and initiatives for the Nemelin myopathy community. So in order to do this, we need your partnership, and you're going to get a chance to learn more about this on this webinar and in the coming months. So as a longtime friend and ally to the foundation building strength, I'm so proud of the things that you've accomplished, and I really look forward to working together. So now I'm going to turn it back over to Mark, and he's going to talk about the mission and vision of the foundation.
Thanks, Sarah, and we really appreciate you taking this role on again. Sarah served as our PCori coordinator in a prior PCori award that we received, and she was great, so glad we could have her back. I guess the story, starts back for me 15 years ago after my daughter was born with Nemelin. And, building the foundation since then with the help of this community has been a real crazy journey. Progress doesn't always occur as quickly as we want, but we've come a long way since 2008. That was when, the care guide didn't even exist. We didn't have a registry. There was no tissue repository in existence. And so AFBS has taken strategic steps to add those foundational resources. So your voice, again, shapes the NM future. And so that's why these webinars are important today. It's only because of this community that we've made it this far. I really want to tell you a quick story. Lynn, if you can go back one more slide quick, tell you a quick story about a researcher that for about 10 years ago that we had a communication with. They submitted what seemed like a very important, but very scientific grant. And when we turned back to that researcher and we asked, how will this research actually serve a member of our NM community? How will it make the community member feel an impact? That researcher could not answer the question. So we actually refused to fund that award. Since that time in 10 years, we've actually rewarded that person 3 different times with funds because they were able to tie the research directly to an impact on the community. So I just want to let you know that your voice is important. And that story has been told again, I mentioned earlier, historically over the years, that has a strong impact in the way we guide researchers to actually focus on our community problems. Okay, thank you, Lynn. So this is a bit subjective, but it is my and my team's view of a picture of how the next 4 years look. Thanks, Lynn. Kate, sorry. This current year, we have a lot going on. We have a very important project to help us understand the effects of gene replacement on CRISPR in NM animal models. We should know that by the end of the year. And we know that CRISPR is an important technology that we understand if it can have an impact in those with Nemorin myopathy. We will probably understand by the end of this year which medicines may help the Neb zebrafish muscles to be more active. We also hope in this year to actually double the number of people in our international registry. By next year, we hope to have 2 companies of industry, drug companies and/or muscle discovery companies that are actually testing NM human tissue in their labs and starting to use their previously created compounds to test the impact. So that's just in 1 year. In 2024, which may be a bit hopeful, but definitely by 2025, we hope to complete a natural history study, which will definitely need your involvement. That work will put us in position to be ready for clinical trials. Hopefully by 2005, we'll be able to identify and start testing an actual treatment. And in 2026, it's my hope that we've launched a clinical trial with an industry partner on a supportive treatment. And we'll also finalize the discovery of a genetic repair for a curative mechanism. Okay, at this point, thank you for taking that in, and love to hear your feedback and any influence you have later. But I'm going to hand it over to Stacy to review the important results of the survey.
Thank you, Mark. Hello, everyone. Thank you for joining us today. My name is Stacy Cassette, and I have a daughter with Nemorin myopathy. I also proudly serve as the AFBS program coordinator and data security officer, but I also work in research as the manager of the Congenital Muscle Disease Tissue Repository, which is located at the Lawler Laboratory. For those who don't know, the tissue repository is where we store patient tissue samples, thereby making those samples available for research use by our scientific community. I'm excited to give you a very brief overview of some results from the patient-centered perspective survey that our Nemorin myopathy community members completed just recently. The survey was developed over the course of December and January with input from about 10 of our community members who represented the diversity of our community, and it was launched on January 31st. It is called Tell Us About You, NM Community. We had at least 108 individuals take the survey so far, and I say at least because the survey is still open as I speak. To give you a little bit of a picture of who our survey respondents were, 28% of our survey respondents are affected with NM. 2% of respondents lost a family member to NM. And 70% of respondents are family members of someone living with NM. The results showed us that the NEB gene variants were represented by 48%, ACTA1 gene variants represented by 26%. And that coincides with what we would expect from a well-represented cohort. So we were very happy to see that. Another 5 NM-related genes were represented by other survey respondents, and we did have some who could not provide genetic information. Our survey respondents described themselves or their family member as being affected in the following ways. 36% reported a mild presentation, 41% moderate, and 23% reported a severe presentation. Next slide, please. The survey itself was quite comprehensive and had just over 20 question items for us to learn more about our community members and the perspectives. And while I don't have time to go over everything in this short time, I'd like to just share a few examples of questions that were posed to our community members and how people responded. So one item asked, what are the most important services or supports AFBS could offer to the NM community in order of importance? The number one item that our community members ranked was opportunities for me to help researchers develop a treatment cure. The other items I listed there were ranked below that, such as research updates in video webinar format, clinical information, hosting conferences, social events, and financial aid. When asked what topics community members are most interested in, the number one topic chosen by 85% of respondents was research and its prospects for medical treatment. That result really shows us what most of us agree on. Research and treatment development is a top priority. And just so you know, that topic of research and treatment stood out from all other important topics that could have been chosen on this survey by our community members. Things like technological aids, meeting others who are similar, accommodations at school, and so forth. So that really spoke to us loudly that research and treatment is a big priority for our community. Next slide, please. Oh, I'm sorry, you're already there. So we were interested in what our community members valued most. So we had asked and provided 12 options for people to rate the importance of what treatment goals would have the most meaning in their lives. And I've summarized just the top 5 treatment goals as chosen by our survey respondents here. So the number one treatment goal was chosen by our community members as any functional improvement. The second, better use of lower body for transitioning, walking, standing, climbing stairs. The third was increasing life expectancy. Fourth, reducing dependency on people, programs, equipment. And fifth, living without mental health issues related to NM. Now, since we understand that it is our community of affected people and families that can make the biggest impact in advancing patient-centered research, we also asked in the survey who would like to partner with AFBS and our scientists in the mission to find meaningful treatments and a cure for Nemaline myopathy. And the results I have shown here. 45% agreed to commit their time for 18 months. 47% said they would like to learn more before committing their time. And all of these people will be contacted by AFBS staff to answer questions they may have and explain the next steps once the survey has closed. If you haven't taken the survey yet, it is still open, at least through this weekend. So go ahead and find the link. It's been shared on social media. It's been sent to individuals on the emailing list as well. Or if you have trouble finding the link, you can just contact one of us and we'll send it to you. So that concludes my portion of the talk for today, but I'll be on here until the end if anyone has questions for me. And I think I'm handing it over to Gus next.
Hello, community. I have the pleasure to introduce to you Dr. Alan Becks. He will give us a brief overview of the current understanding of the causes and consequences of Nemaline myopathy, some clinical characteristics, and the current research portfolio aiming at finding medical treatments. Dr. Alan Becks is an expert on myopathies in general and Nemaline myopathy in particular. He's a longtime advisor and a really good friend to AFBS in everything related to research from discovery to the translational path towards medical treatments. His contribution is too extensive to mention in this short webinar, so I invite you all to check his bio and lab websites. I will add the links in the chat. He is currently the director of the Manton Center for Orphan Disease Research at Boston Children's Hospital. And he will be accompanied by Casey Genetti. She's a project manager and genetic counselor in Dr. Beck's lab. The stage is all yours, Alan and Casey. Thank you.
Now, thank you very much, Gus, and thank you, everybody. It's wonderful to see everybody here. I see a number of names I recognize, old friends, as well as some new names. And I look forward to seeing everybody and meeting everybody who I don't already know, hopefully this summer at the QRCMD conference, which I think I will hear a little bit more about later this morning. So this is the first of 10 PCori funded seminars, as Mark mentioned at the outset. And so I thought that today I would just give you guys a brief overview of the scientific and medical aspects of Nemaline myopathy. As you'll see from some of the research that we've been doing, in fact, what we find from interacting with you guys, with patient community, with speaking with physicians, with reviewing medical records, is very parallel to what you folks are all reporting in your patient-reported surveys. But just at the outset, I want to emphasize the importance of what we're doing today, what you're doing and participating in, which is making sure that the patient voice is heard in all of this. Because oftentimes your perspective is different from the perspective of a physician or a researcher who maybe sees your child or yourself only briefly, so a couple times a year. And so PCori as an organization has been formed really to ensure that the patient community and the patient voice is heard, both in helping to understand these rare conditions like Nemaline myopathy, and most importantly, in setting goals for appropriate treatments so that what we do in the research community is actually really going to help the people who need it most. So to start, Nemaline myopathy, it is a name given to a group of muscle conditions based on initially what's seen under the microscope. So a lot of diseases before we really knew what the causes were basically named on some aspect that seemed to be characteristic. So either a clinical aspect like muscular dystrophy, which means muscle not working properly, or in this case, Nemaline myopathy refers to the word Nemaline, as you'll see in a minute, corresponds to structures seen under the microscope. So if you look at the lower left-hand corner here, the pictures, there we go, thank you. That blue picture in the lower left-hand corner represents what the muscle looks like under the light microscope from an individual with what we call Nemaline myopathy. It's stained with a special stain called Gamori trichrome, which stains the fibers blue or green, blue-green. But you see these dark blue dots within those fibers, and those are the so-called Nemaline rods or Nemaline bodies. And just to the right of that, the gray image is a picture under the electron microscope. Instead of being in cross-section, now the muscle fibers are shown lengthwise. And the very dark structures above that round structure, which is the nucleus, Those dark structures are the so-called Nemelin rods, and you can see they appear somewhat rod-like here. Sometimes they're rounder or more circular. So you'll hear them referred to as Nemelin bodies or Nemelin rods. It's the same thing. The word comes from nema, is the Greek word for thread. So nemeline is thread-like, and that's how they were described. So the condition then is really defined on the basis of skeletal muscle weakness together with this finding under the microscope. And as it turns out, if you go, for example, to your local Muscular Dystrophy Association clinic, this is one of the most common of the congenital myopathies. So the word name congenital myopathy, just very descriptive, simply means myo is muscle, pathy or pathology is disease, and congenital means you're born with it. So it's a muscle disease that you're born with. You've also heard, and many of you may have been told at some point in your initial diagnostic odyssey, that perhaps you or your child might have had something called muscular dystrophy. The muscular dystrophy is somewhat more common and is something that's been in the newspapers a lot. And so there's often confusion between muscular dystrophy and the so-called congenital myopathies. And so sometimes I'll refer to the group of conditions that Nemelin myopathy belongs to as the non-dystrophic myopathies. And the distinction is that in the muscular dystrophies, the muscle is capable of contracting properly, but the muscle, because of defects in its membrane and other aspects, dies over time, and it regenerates and it dies, and it regenerates and then it dies again. And over time, it loses the ability to regenerate. And so patients with muscular dystrophy have severely progressive courses. They get weaker in a very characteristic pattern over time. In contrast, the congenital myopathies are relatively speaking non-progressive. That doesn't mean that you don't change over time, that individuals as they age don't get weaker, but it's not this rapid and relentlessly progressive course that you see with the muscular dystrophies. And so as I'll show you in a few minutes, the reason for that is that the basis for weakness in the Nemelin myopathy is not that the muscle is dying. In fact, the muscle is quite stable and stays around for a lifetime. However, the underlying defect is in the ability of the muscle to contract, is in the actual contractile apparatus that allows the muscle to contract. So these are primary muscle diseases. And as an entity, as a group, the Nemelin myopathies are thought to roughly have an incidence of about 1 in 50,000 births. So this is a rough number, and depending on who's doing the counting, it may vary somewhat, but roughly 1 in 50,000 births. So that makes it actually fairly common with, and as you know, you've got a nice robust community. So it's not such a rare disease after all. And I mentioned non or slowly progressive. Muscle weakness and hypotonia. Hypotonia simply means floppiness. And babies that are born weak or floppy are called hypotonic. And it's quite mild. So you see pictures here of 5 individuals. All of them have a form of Nemelin myopathy. They've all given us permission to show their pictures. And it ranges from this baby at the lower left there, who sadly had a condition so severe they never moved in utero, and when they were born, came out with these massive contractures, and sadly that child didn't survive the first few days of life. In contrast, you can see several children who are standing, or a girl who is obviously has a wheelchair behind her and has a tracheostomy, but they have somewhat less severe conditions. And then finally, this man on the bicycle at the upper left had a condition you can see if you look at, for example, his arms or his lower legs, there's reduced muscle mass, but he had a condition, in fact, that affected just some of his muscle fibers. In fact, his fast muscle fibers that are used for sprinting. And so he was strong enough, in fact, to do endurance bicycle racing, and he's shown here doing that. So the point here is that this is an extremely variable condition clinically, ranging from unfortunately, some infants and young children who don't survive early childhood, all the way up to people who grow up, have families, become grandparents, and everything in between. So the next slide shows a little bit more about the pathology and the characteristic findings. So here on the left, again in Gomori trichrome, are shown 2 different pictures. And at the top, you see tiny rod bodies inside muscle fibers that are somewhat varying in size. You can see the diameter of these circular structures varies. But not all the fibers have nemaline rods, and the nemaline rods tend to be relatively small. In contrast, at the bottom, virtually every fiber is filled with large numbers of very large rods. So looking at this, you might say, well, for example, maybe the patient whose muscle is shown at the bottom left in panel C has a more severe condition, and maybe the patient in panel B has a somewhat milder condition because there are fewer nemaline rods. But in fact, if you advance the slide on the right, you'll see this is the picture in the electron microscope looking at the structure of the muscle and cross-section. And in the lower right, you'll see very organized zebra stripe pattern of muscle with a number of large rod bodies in between. But that highly organized structure actually is intact muscle fiber capable of contracting. Whereas at the upper right-hand corner, you'll see fibers that are extremely disorganized. That one big circular or oval-shaped structure at the top there essentially has no organized myofibrils or contractile apparatus. And so the top two panels then came from a child who was extremely weak and unfortunately didn't survive infancy. So the picture then clinically is variable. And when we look into the microscope, it's variable as well. The next slide. So then what is the genetic basis for this? So this illustrates what we know about some of the genes. And this is work that my lab and many of our scientific collaborators have contributed to over the years, and that AFBS has helped to fund over the years as well. And shown here within this oval are the gene symbols for a variety of genes that can be mutated to cause nemaline myopathy. So I think many of you will recognize genes here that you know that you carry or that your children carry. So for example, NEB stands for the nebulin gene. And that symbol is the largest because it turns out that nebulin mutations account for the greatest proportion of patients with nemaline myopathy, roughly 45 to 50%. Mutations of the skeletal muscle actin gene, or ACTA1, are the next most common cause of nemaline myopathy, and so that's the next largest gene symbol. Following that, the tropomyosins, tropomyosin 3, tropomyosin 2 are common. And then there's a group of less common genes, for example, KLHL40, KLHL41, and so on, that can also be mutated to cause nemaline myopathy. And Stacy, a little while ago, showed you a little bit about data on the relative proportions of patients who have mutations in each of these different genes. Casey Giannetti in a few minutes will show you the results from our data bank from patients like you who've enrolled with us over the years and shared your medical records. And you'll see that it's actually, these numbers match up really very nicely. So I mentioned that the diagnosis of nemaline myopathy is based on what the muscle looks like under the microscope. What's shown here now are a group of conditions called core myopathies. I'm not going to get into those except to say that instead of nemaline something we call a core, or which represents a breakdown in part of the muscle structure, and RYR1 or ryanodine receptor is the most common cause of that. But you'll see some overlap. And so, for example, occasionally patients with a mutation in the actin gene can cause those core myopathies. Central nuclear myopathies are another group. And then finally, the congenital fiber type disproportion, if you advance one more time, is yet another finding by pathology. And you can see that, for example, mutations in tropomyosin 2 can appear, or tropomyosin 3 can appear to be CFTD. So the point I want to make by this is that sometimes the clinical pathological diagnosis that's given to somebody based on the way the muscle looks under the microscope doesn't always match with the genetic diagnosis. But our understanding of the underlying genetic basis is what's really critical in order to help us think about how we develop therapies. So what do these genes do? Well, this is a diagram of muscle that some of you may have seen. And in fact, I believe there's a YouTube video that we've created based on an old Flash animation which I'm hopeful one of the organizers will put into the chat at some point, that illustrates how muscle contracts and talks a little bit about this. But just to very briefly orient you, in the center here, the circle that's labeled sarcomere represents the contractile apparatus of skeletal muscle. And muscle contracts by these two vertical gray bars labeled Z-band, getting pulled closer together. And they're pulled by these red structures in the middle that are labeled the myosin thick filament. And the myosin thick filaments pull on these narrow gray structures labeled actin thin filaments to actually bring the Z lines closer together. Well, as it turns out, if you look at the bottom of this diagram, where this actin thin filament is labeled with a series of proteins, you'll recognize there's nebulin, troponin complex, tropomyosin, and actin. These are all the structural components of this thin filament. And as it turns out, the genes I was showing you in the last slide encode the proteins that make up this structure. So now we can begin to understand actually how the weakness arises. In fact, if you have a defect in these actin, the gray balls, or if you have a defect in nebulin, then the thin filament structure itself is going to be abnormal, and this entire contractile apparatus isn't going to work properly. So to summarize in the next slide, this is something which has been interesting from a scientific point of view, but frustrating really to some extent because it's making gross generalizations difficult and has required that we think about different things in different ways. But essentially, Nemaline myopathy is a very variable or heterogeneous condition. As I showed you, the clinical heterogeneity, as you know, varies widely from very severely affected individuals to individuals who have significant disabilities, but yet, for example, may walk or not have too much respiratory weakness for much of their life. Pathologically, there's a lot of variation. And then on the genetic basis, there are multiple inheritance patterns. So some genes are inherited in a dominant fashion. You only need one abnormal copy of the gene. Others are recessive, whereby you need two abnormal copies, one from each parent. Now, of course, the multiple genes, the individual mutations that people carry can vary. And in extreme cases, I didn't talk about this yet, but for example, although most patients with an actin mutation have a single abnormal copy, and so therefore what we call dominant inheritance, some of the mutations in fact can be recessive. So I'm going to turn it over to Casey Giannetti now to tell you about a study that we're just in the process of doing now and getting close to completing, looking at our medical records for, we have about 300 individuals who've enrolled with our research study over the years. This study has been supported over time by the AFBS and is allowing us to kind of really try to put numbers and correlate some of the clinical findings together with some of the genes. So Casey, if you'd like to take the next couple of slides, that would be great.
Sure, thanks, Alan. Great to be here and to see so many names that I recognize as well. And hopefully can get to know some of you who I haven't met yet in the future. So as Alan mentioned, starting back, I guess, in 1990, when we started collecting our patients with different congenital myopathies, we now have amassed about 300 and growing individuals with Nemaline myopathy. It kind of represents our largest cohort of patients that we have here in the Becks lab. And this table here just shows a breakdown of 219 of those Nemaline myopathy patients who have a known genetic diagnosis. And one of the issues we'll talk about later is we still have a pretty significant portion of patients who have a clinical diagnosis, so perhaps from a muscle biopsy of Nemaline myopathy, but despite having genetic testing, we still don't know their actual genetic diagnosis. So whether that's just a tricky genetic variant to find by our current technologies, or perhaps even more likely, newer genes that we just haven't yet been able to establish. But you'll see the breakdown of our particular cohort here. And just like the response from the surveys that were completed recently, the largest group of patients in our cohort have mutations in NEB, or the nebulin gene. It's about 35%, followed by ACTA1, close to 30, And then TPM3 and TPM2 are the next two. Genes like LMOD3 are TNNT1 and the KLHL genes make up just smaller numbers, as well as a couple of the other genes shown on Alan's slide that we don't yet have patients in our cohort. But these numbers are growing and changing every day as we're getting new patients enrolled who already have genetic diagnoses, and also through the testing that we do as part of our lab for patients who don't yet have a genetic diagnosis. And as you'll see in the numbers as we go down on this chart, every gene has variability within it. So it's not just variability gene to gene. Even within each gene, we see ranges from some of the more severe congenital presentations all the way to what we might describe as adult onset. And so you can see that breakdown there. Go to the next slide. Just to drill down a little bit further, focusing here just for display purposes on our three most common genotypes, so the Nebulin, ACTA1, and TPM3. You can see that the variability, here in things like feeding support. So some of the information that we're pulling from these medical records and from interviews with our patients are things like ambulation, feeding support, respiratory status, history of delays and motor delays. So with all three of these genes, as well as most of the genes, we see pretty significant motor delays in the population. So over 3/4 in all three of these genes having some type of delay, say in crawling or in walking. With feeding support, you can start to see a little bit of the differences between our genes, where, perhaps for the TPM3, it's a smaller number who rely on a G-tube or NG tube compared to our Nebulin or ACTA1 individuals. You can head to the next slide. And then here we're showing our respiratory status. So something that we know from patients, obviously, is a very important piece of their kind of every day-to-day clinical presentation, and perhaps to, something throughout life is one of the most important aspects that they'd like to see improvements in. And so we can see for our Nebulin and ACTA1 cases, we have a higher percentage of invasive ventilation. So perhaps a ventilator or a trach compared to TPM3, where we're seeing a higher rate of non-invasive ventilation, like a CPAP or BiPAP. And then also, our largest contingency here is actually those who don't require any respiratory support at all. And so these are really important aspects of the condition that we are collecting through these records and natural history over time, which we'll talk a little bit more about in kind of our coming slides, why understanding this natural history is so important as we move into therapy development and clinical trials. You can go to the next slide. I'm going to throw that back to you, Alan, for this one.
All right, thank you. And I would just say, to avoid any confusion, I think Microsoft Office was looking out for us, and the TPM3 was auto-corrected to TMP. I think they thought we were trying to say temporary or something, abbreviate that. But it is TPM3, not TMP3.
Yes.
So just saw that now. So, why do we care about natural history? Why do we want to understand? Well, from your perspective, you want to know what to expect for the future. But if we hope to develop treatments, then we need to be able to know that those treatments are going to be effective, and that we need to be able to measure the beneficial effect of a treatment. We're going to probably have an opportunity, or maybe many opportunities, to talk in detail about treatments in upcoming webinars. But just to kind of whet your appetite and give a quick introduction, there are approaches to treatment that involve small molecules or drugs, and there are a number of studies, many of which have been funded over time by AFBS, that, for example, are looking at drugs that improve the muscle's efficiency of contraction, that operate, for example, in patients who have mutations in the Nebulin gene. In the next slide, my group and others have been working on approaches to gene replacement therapy. Not every gene that causes neuromyopathy is necessarily amenable to that. So, for example, The nebulin gene is too large to fit into the viral vectors that currently we use for gene therapy. But the actin gene is actually small enough to fit. There are challenges that we have to think about. So for example, actin mutations are dominant. So having one mutant copy actually damages the cell. So we have to think about how do we remove the effects of the mutant copy and add back good versions. And then in the third slide here, there are also approaches to genetic engineering. Some of you may have heard of CRISPR-Cas, or there's a new approach called prime editing. These are all approaches that allow us to go in and actually alter specific locations within the DNA. Those could be very useful approaches for things like Nemelin myopathy caused by mutations in the nebulin gene, or some of the more rare conditions. And I'm not going to get into it today, but there's now, many of you may have heard of this concept of personalized medicine. And so individuals who have mutations in KLHL40, for example, TPM2 or TPM3, some of the more rare versions of Nemelin myopathy, it may be that we'll be able to utilize some of the recent advances to develop approaches for that. Well, the next slide talks about, what do we need to do if we're successful in the laboratory? What do we need to do to translate this to actual clinical studies and eventually treatments that can be available to patients and families? And that's really, I think, what the PCori program is aimed at supporting. So as you just saw, through surveys like the one that Stacy described that you recently may have participated in, as well as enrollment in our research studies like the one that Casey was talking about in my lab. It's very important to develop registries. CureCMD, in collaboration with AFBS, has a registry, which I encourage everybody to register with. And this is important, not just to help us all communicate with each other, but also to provide evidence to companies and to funders like the Muscular Dystrophy Association, AFBS, potential donors and supporters of AFBS, to really demonstrate that there's a major unmet need out there, that there are patients out there that if we develop a treatment, that there are people who can benefit from that and who need it. Another important area that my group is very active in is improving the genetic diagnosis. So how do we find mutations in the genes we've been talking about more easily and efficiently? And how do we discover some of the genes that we don't know about yet? So there's probably still a small handful of genes out there that we haven't been able to identify, and yet they likely also cause Nemelin myopathy. The other aspect that, again, PCori really focuses on is what's so-called clinical trial readiness. And this is basically the concept that we have enough necessary information to actually design a clinical trial of a potential new drug. And so as labs like mine and others are developing potential new treatments, we need to have the information available so that physicians can design studies that would accurately measure this. And so what Stacy and Casey have told you about, the so-called natural history studies, have been are critical in order for us to know what normally happens if we don't treat the condition. We need to be able to measure things. So for example, muscle strength, breathing ability, ability to walk, and so on, so that we can measure improvements in that. And then to some extent, looking through the tissue repository in the Lawler Lab at the Medical College of Wisconsin, also supported by AFBS, we have the opportunity to search for biomarkers. So biochemical features of muscle or blood that are specific to the disease that we can then ask, do they get improved following a treatment? So the last slide that I'm going to present, and then we want to leave time for robust discussion and questions on the next slide. Just kind of, I think, for me, summarizes, the importance of what we're doing here today and who's here and what are we doing. So critically, we can't consider effective drug development without the involvement of you guys, without the patient community. So you help us define the unmet need, you partner, and you're available and willing, some individuals, to enroll in and volunteer for medical studies. Casey and I represent the academic community. We're here to educate and to work with you to develop our better understanding and develop treatments. Once we get beyond a certain point, though, to really support a clinical trial, oftentimes we'll be partnering with biotechnology startup companies or large pharmaceutical companies. And Mark mentioned AFBS is working together with industry to help bring their resources to bear on this. But then finally, if you forward the slide one more time, foundations and patient advocacy organizations such as yours, if you forward the slide, really represent the seat of this tripod. So the stool, you need 3 legs for the stool, and AFBS really kind of brings everybody together. So with that, I'll end. Probably apologize for taking more time than I think we probably anticipated, but I want to leave time for questions, and so I'll turn it over to Patty Mitchell. Thank you.
Thanks, Alan. Hi, everybody. It's so great to see so many names that I know here. For those of you who don't know me, my name is Patty Mitchell. As Mark mentioned, I'm a volunteer board member with AFBS. I also have a daughter that has Nemaline myopathy. And so just going to sum it up, we're excited. We got this PCori grant. It's basically $250,000 over the next 2 years. In addition, we've got the Chan Zuckerberg grant, which is around $600,000 over 3 years, as well as several large donations, some of which are international. We have greater funding now than we've ever had. We have a very aggressive timeline of things we want to achieve. The whole purpose of the PCori grant is to engage the community because we can do more together. And the researchers, we know, because we have to advocate for ourselves constantly as parents and people living with NM, the researchers can do more when we advocate for ourselves with them, when we give them input, when we're involved with them. And so that's really the whole point of this PCori grant. The survey asked who was interested in participating. Many of you said you were. Others said you wanted more information. Those who wanted more information, we will be in touch within the next 2 weeks. But overall, what we're looking for and what is stipulated in this grant is that we want to find community members who will do 3 things. 1, they will participate in the webinars. We're going to have 10, including this one, over the next 12 to 18 months. 2, you'll be a voice for the NM community. You'll listen, you'll ask questions, you'll provide feedback. And 3, if you could join us at SciFam, either live, which is our scientific and family conference, which we have a great partnership with Cure CMD, so we can leverage expenses. It's a great event. We're going to have live involvement as well as a virtual involvement. Then you can really represent the face of NM for our researchers. And because your time is valuable, this grant does authorize us to compensate you for your time and for your involvement in this effort. So we're authorized to compensate those who commit, which we have a limited number, but it's a fairly large number that we can have down as committed, would be compensated in the amount of $300 for each of the 2 years that they were involved. And so in summary, I'd like to thank you for your time. And at this point, we'd like to turn it over for questions. So if you do have a question, if you could raise your hand or type your question in the chat, we would appreciate it. Thanks for making time.
Okay, so who do we have moderating questions here? Gus, is that you or?
That's me. Sorry, I couldn't get my mute button. Hi, this is Terry. As they introduced me, I'm a grant consultant for AFBS, and looks like we have a question from Casey. So this might be good for Gus or Alan. I was told in an informal conversation that adults or older children may not be good candidates for CRISPR or gene therapies. Younger children will have a better response, and older children or adults may not be included in preliminary clinical trials. Any validity in this, and what's your take on the age factor?
Alan?
It looks like Alan was frozen. Okay.
Apologies, everybody. My internet just croaked. I'm back.
Alan, do you want me to repeat the question, or can you see it in the Q&A?
Yeah, okay.
So
Age. So,
Alan, I think we better
are you
start
guys-
again. You're still in a bad spot.
Okay, I can take at least part of the question, and of course, Alan can add maybe later. This is, of course, still in discussion. There are gene therapies, and I mean, we, even if Nemaline myopathy is not an advanced fast as dystrophies, the progression and the accumulation of muscles that are involved or weaker, of course, as you age, that increase, and so the gene therapies or gene editing will probably work a little bit less than in younger children. But for the clinical trials, we have to do the natural history study first and see what age ranges we can define outcome measures and these treatments be amenable. And the other thing is some complications with the vectors that are used now for gene therapies or gene editing, this AAV, adeno-associated virus vectors, that as probably you also heard that these are one injection or one time treatment, because once that you provide the new gene or the modified gene with these vectors, you create some immunity for that vector, so you cannot do a second injection at this point of the technology. And the other thing is that probably the population get exposed to AAVs and have some immunity before the treatment. So as you age, you can be more resistant to the AAV injection than younger children. But these are all in debate and being very heavily studied at the moment. Alan,
Casey, can I just, before Alan, you insert your response as well, I just want to make sure that Casey knows it's an important question. Obviously, we've got a lot more to learn in this area, and we'll have these 10 webinar series to interact with more and more researchers that will be able to sort of help us sort of get a little bit more important answer to your question. But I think the question itself is of value, because it says, how are we going to guide research to treat the older adults? And that's really important as well. So Alan, I'd love to hear if you have anything in addition to add what Gus said.
Sure. I'll just say briefly that don't despair, because one of the hallmarks of congenital myopathies is that they are less progressive, and everybody's going to be different, and everybody's going to change throughout their lives. There are some aspects in particular for muscular dystrophy. The muscle is lost over time, and so then so that's the basis for a lot of people saying that in those diseases it's really critical to treat at an early age. But for Nemaline myopathy, the more that you can maintain your function through physical therapy, maintain flexibility, passive range of movement, avoid developing contractures, maintain good respiratory health, the better a candidate you will be for treatment over time. And there's no hard and fast rule about this. And I should just say also that the rule doesn't apply specifically just to CRISPR or just to gene therapy or just to drug. It's going to be a little bit different potentially based on what the modality. But there's nothing that we know of that today that we would say precludes an older individual from benefiting from any of the treatments that we're trying to develop. I also see Anna Halgren. Hi, Anna. It's nice to see or read your writing. Is asking about studies in other countries. And of course, Anna, I know you're in Europe. and we have a really nice international community. So while AFBS is led by Mark and his colleagues in California, here in the United States. In fact, if you look at the website or speak later on with Gustavo or Mark, you'll find that many of the grants have gone, for example, to investigators in Amsterdam. I've been working with investigators in Sweden, investigators in Australia, and so around the world. And so this is not just the United States, this is an international initiative. Mark or Gus, I don't know if you want to expand on that at all, but.
Well, that's perfect. And again, we're not restricted. We're not restricting at all the participants that we'd like to have join us. So we encourage international participation in these webinars. So yes, please continue to spread the word, get more people involved. And we understand that we have something to learn from every single person in the community.
So we have a question about genetic testing at an older age. Well, AFBS does help, but in fact, if you maybe Casey, you want to take this question?
Sure. I'm a genetic counselor, so I deal with a lot of genetic testing questions and logistics. So feel free to contact me after this, and we can talk about it further. It's different in every situation. There's clinical testing that's available, and there are laboratories that actually offer assistance when insurance might not cover a panel. And then we also have our research lab, which can help out with research testing, at least to look at a large panel of genes. And it often needs to be double checked by a clinical lab, but for a very specific indication, if we can find it first in the research basis. So lots of options. It's different for every case. So feel free to reach out and I can help move you forward in terms of what would be the next best steps.
Yeah, and if you're interested, you'd be welcome to enroll in our research study. Our approach is to first work with your physicians to see if we can arrange clinical testing that would be paid by your insurance, simply because that would be the fastest and most efficient approach. But then through the support from AFBS and other groups, we have the abilities to do research-based testing, which may take a little bit longer, but gives you just as accurate an answer. And in fact, we often then arrange for a clinical confirmation afterwards in collaboration with anybody who enrolls with us.
And again, that question was, as an older person with NM, I've never had genetic testing. Will AFBS look at help in funding and organizing this for those who would like to know this?
Yeah.
Anyone
Thank you,
can
Terry.
see the question? Sure.
I think we'd like to understand more about, the need and demand for the communities. That's why these webinars are important. And I understand, Casey, that the cost has come down quite a bit from whole genome sequencing years ago to what they do cost today. And I just want to say hi to Melissa too, because I haven't seen her in years. So Casey, do you know the cost has come down significantly, right?
Yes, absolutely. The cost has come down, the availability has gone up. So we have lots of commercial labs now offering testing with either panels or some of that expanded whole genome or whole exome. But in all ways, it's become much more accessible, which is fantastic. And we've seen a really large boom in terms of the number of patients who are even able to be diagnosed before having a muscle biopsy, which wasn't even possible just 10 years ago. So it's an exciting time.
Yeah, and we really encourage for everybody to look for its genetic confirmation, because it will be important for the natural history studies for selecting and recruiting the cohorts for clinical trials and treatments.
And I know that Stacy's on the line. She can probably answer this question, but I know, and Rachel Alvarez also is a participant, that we have at least a minimum of 70 people in our registry that have not been confirmed genetically. And so it's definitely a need. How can we give information to the researchers on the cohort that we have if we can't identify them? So we'd love to get more people registered. We'd love to get more people confirmed. And that's a commitment from a foundation building strength.
For any of those 70 people, if they are enrolled with our lab or would like to enroll with our lab, we can certainly facilitate the process. So.
now that Alan mentioned that, we encourage you to register in both, in the CMDIR lab.
Any
Well,
other
so-
questions? Oh, sorry, go ahead. Mark.
Yeah, no, I was gonna say it's 11:03.
Yeah, I was just going to say, any questions about the great presentation from Alan and Casey, or participation in the PCori project over the next 18 months, 2 years? As Patty said, or Sarah said, we'll be reaching out to families who in the survey have indicated that they are open to participating. And we're really looking forward to working with you.
I think there's one more question from Joanna, and I think I can answer that. The foundation has been fortunate not only to receive these several awards, but to actually have some family foundations as well as individual contributors that have really increased their impact. And so I think the amount of dollars that's been coming into the foundation will have probably quadrupled in the last several years. And so the amount coming in and the momentum we have in funding has been amazing. Building this community may be another resource to be able to continue to build our database and connections to contributors. So we hope to do that. We have important new board members, including the Machado families, which I've seen in the participant group here, that have strongly contributed to new research. And so we're very, very excited about the momentum, about how much money we have in the bank. Right now, we are very well funded, and we're waiting for the next important projects. We are trying to reserve some base of commitment for the natural history work, because we know that is really important to get us to clinical trial readiness. Thank you for that question, Joanna.
And I should add that one of the aims for AFBS is engage with pharmaceutical companies, because we will need that leg and that partnership, of course, to move treatments forward.
Very true.
And we work heavily in trying to convince or bring pharmaceutical companies to work with us and help develop treatments.
Alan, I have a question for you. So individuals, or Casey, individuals who are involved in your research program and you do identify a genetic variant that's causal to their form of disease, do you provide the family with a genetic confirmation report at that time, or do you just hook them up with a clinical testing lab, and then that lab would be the one to generate the report?
Thanks, Stacey. That's a great question. Because of the regulations we have here in the States for research studies, we do have to do that extra step of having a clinical lab confirm our findings. So what we can do is generally let a family know that we found something that we think is diagnostic for them. And then generally we can work with their providers, be it a neurologist or a primary care, really anyone who's part of their team, to then have that testing ordered. We don't need to redo the large scale testing we did. Generally, we can tell that clinical lab the exact genetic change or changes to look for. So it's a much faster and less expensive test. But since a research lab doesn't have those same quality controls as a clinical lab, we do need to have that extra step to get an official report.
In the genetic process, Casey and Alan, are muscle biopsies still commonplace? Or are we jumping right to whole genome sequencing sooner these days?
It's a mix, I would say. And sometimes we still need both. We're seeing certainly many more cases where genetic testing is being ordered, especially for young kids, before a muscle biopsy. But still, a muscle biopsy can be a gold standard in the diagnostic process, particularly if a genetic result is unclear. So if we have what many of you may know of as something called a variant of uncertain significance, or perhaps, on our first tries of genetic testing, no real leads at all, the muscle biopsies can be very helpful to steer us in the right direction. Or do additional research, which some of our technologies that Alan could talk more about too.
And so there's no rules, there's no requirement for muscle biopsy necessarily, but in many cases, in some cases at least, it can be helpful. And it used to be the basis for the diagnosis. I should say that for anybody who has had a diagnostic biopsy, one aspect of contributing to the research is making that biopsy available. Once the clinical testing is complete, there's often a little bit of muscle left over. So that's an option on the informed consent forms for both the biobank in Wisconsin and our group as well.
Another question. If a person has TPM2 and wants to try individual treatment, what do they have to do?
That's a great question. As much as possible, as we develop treatments in the research field, we try to do things that are going to impact as many people as possible. Whether or not an individual treatment is possible or available is going to depend on the particular mutation and on the biology. And right now, we and others are thinking about other ways of, more general ways, but I think engaging with researchers, speaking with people who understand, in your case, tropomyosin 2, who understand that particular mutation, it really needs somebody to come up with the bright idea for exactly how to do that. But just in general terms, there are a lot more opportunities to think about those approaches than we knew about a few years ago, for example.
Yeah, so I think right now, Anna, there are, reactive treatments. What are your symptoms? How can we respond to those treatments? And I'd be happy to talk with you offline about what I think the best science is around Nemelin research. And we hope to have more sort of therapeutic specialists that will talk through, best care and best practice as these webinars continue. Leticia, thank you for your note in the chat. Just want to say thank you to everyone that came today. Really happy that we had the participation that we've had, and very, very grateful for this community and what we can do together. I'll just ask for any other comments from the group here. Anyone else have any closing comments? Patty, you want to give us one last cheer?
I guess I'm the cheerleader. They think I'm really positive or something. I don't know. It's the Midwest. What can I say? I've been involved over 10 years. I've never seen the momentum like we have now. We have a great team, and we're very excited to work with all of you. And so excited to accomplish some great things together. Thank you, everybody.
And really quick, I hate to close on a last question. Is there any treatment for TNNT1?
Okay, that's something my group is working on, and there is a group in Pennsylvania working on it because, as you may know, there are a number of Amish families with mutations in that gene. I've been focused a little bit more on the dominant form, but yes, if anybody's interested to reach out and ask us more, I see that Casey has just posted her email address in the chat. Feel free to reach out to her and through her to me, and we can discuss further.
Thanks again, everybody.
Great.
Have a great Saturday.
Great to see everybody. Thank you.
Thanks, Alan.
Bye.
Nice to meet
Thank you.
Thank you all.
Alan.